That's it. Nothing else. I'm not asking for anything else more. A balance. A cell theory, it is, I'm not sure if this is coming out on the screen, so let me darken up the room, even though we're going to lose some nice sunlight. But all organisms are made up of cell. That was the answer to your exam question, last exam. All organisms must be made up of cell. All the way from bacteria to the cockroach, the fungi, the mushroom you eat, they're all made up of cells. The bacteria make you sick, it's made up of cell. I'm not talking about viruses. Viruses are not living organisms. That would be nice essay questions. Why viruses are not living organisms? Because they do not have cell membrane. Some viruses do not have DNA. They have only RNA. Okay? And they do need a cell to replicate. Without a cell, they cannot replicate. So they are not living organisms. Viruses are not living organisms. They need a cell to replicate. Whatever cell it is, a bacteria, human cell, whatever cell, animal cell, plant cell, fungi. That's why viruses are not living organisms. Okay. The cell is the simplest collection of matters that can be alive. And what is alive, that's what Brian wrote in his exam. He said, he gave me the definition of what is alive. Alive, if biology, it means study of life, right? You guys, Mendoza, biology means study of life. And what is life? Life is anything that can multiply. Viruses cannot, without a cell. Viruses cannot multiply without a cell. Anything that can multiply and anything that can do metabolism, both. Multiply and metabolism. Those two are needed for something that can have life. Viruses, they do not do that much of a metabolism that we know of. And we talk about metabolism a little bit later on. Okay, all cells are related by either descended from earlier cells. It means all cells came from a common ancestor. Life came from a common ancestor. That's what most biologists believe. Cells can differentiate substantialities from one another and share common features. So cells, for example, the cells, some of you, I asked the question, I asked what is it that the cells from Mars have to have in common? Some of you wrote down cilia. Well, I have cilia, and the cells you see in the lab, the cells from pond, if I go grab a pond water, some of the cells have cilia as well. That's commonality, but I have DNA, the cells, the cockroach have DNA also. So there are some commonalities, and I asked that in the last exam. And that's what cell theory is. All living organisms must have this commonality among themselves. Then the size of the cells are different. Here is the size of proton, here is the size of a human, and a neuron, an egg, and most of the cells are about 110 to 100 micrometer, and of course bacteria and organelles are much, much smaller. Here is the other shape, something. These are different type of microscopes. They're used to look at things on the microscopes. Again, I'm not gonna say too much about it. But there are two basic types of electron microscope, EM they call them. At Delta College, we do have a program, a certificate program and a 2-year program called electron microscopy program. You go get there, after 2 years you get your degree, and then you can go for NASA or any other big places and make good money. Not too many people, not too many. No other community colleges, not too many. No other community colleges in California have that program. Only Delta has it. There's another one in Wisconsin, I've heard. So there's only 2 community colleges in the United States have the same program that Delta College has, electron microscopy. Okay, so anyhow, you can get a 1-year certificate degree, or you can get a 2-year degree, which is a AA degree, I believe. Scanning electron microscopes is SEM4 focuses on beams of electrons onto the surface of the specimens and providing images that look like a 3D. So they look at, and I'll show you some pictures. Transmission electron microscope, on the other hand, they cut the cell, they take the cell and they cut it so you can see inside of the cell. And magnification is a lot more. Focus on beams of electrons through specimens. TEM transmission are used mainly to study internal structure of the cells. With scanning electron microscopes, you don't see anything inside of the cell. You see outside of the cell, but huge, very huge. That is scanning electron microscope. Cells fractionation, that's what we were talking about. How do you look at the cells? That's another way. Cell fractionation takes cells apart. They break them. They break the cells. That's what it means. They are in a test tube and they pass a rod very, very tight to the test tube, and so the cells break. Separate major organelles, centrifuge them, and then components go. The heavy components go to the bottom, and the light components go to the part. Cell fractionation enables scientists to determine the functions of organelles, biochemistry, and cytology. What does cytology mean? Cyto means what? Cell. And cytology means what? Study of cell. Your prefix suffixes. Some of you did not study that very well. Well, you were confused by haploid. Hal, it means half. Is that right? But I asked for haploid. Function and structures. Here it is. They break the cell. Here are the cells, and they blend them, and then they centrifuge them, and the heavy stuff goes on the bottom, and the light stuff goes on the top. They take the light stuff out, and then the heavy stuff, they grind them again, and then so on and so forth. They get to the bottom. The smallest one were up here, were on top here, and now they're on the bottom of the test. They centrifuge them, so on and so forth. Comparing prokaryotic and eukaryotic cell, that was another question I asked on your list. Is that right? That's the question I'm asking. What are the differences between prokaryote and eukaryote? Basic features of all cells: plasma membrane, semi-fluid substance is called cytosol. So what is cytosol? All of the fluid inside here, not the organelles, the fluid inside here, inside of the cell, the fluid inside of the cell is not actually fluid, it's a salt gel. Remember I talked about that, like a jello? If I put a syringe in here, and take all of that liquid out and put it in a test tube, it would be like a jello. It's not going to be liquid. It's a jello. It's not solid, and it's not liquid. I didn't bring water with me today, but it's not liquid either. Well, I don't want to open up. It's not, well, this is like a jello. Yeah, it's just like this. So cytosol, it's a jello-like. Chromosomes carry genes, of course. Ribosomes make proteins, and plasma membrane or cell membrane, which is right here. If I put a piece of tape during lab practical exam right here, if I put a piece of tape during lab exam, what is the name of structure? What is that? Plasma membrane or cell membrane. All of this around, there is no cell wall in here. This is an animal cell. Animal cells do not have cell wall. They have cell membrane. Okay? Do I make sense? That's the cell membrane. Allows sufficient passage of oxygen, nutrients, waste. So this is the border of the cell. This is the border of the cell, allows molecules in and outside of the cell, right here. Okay? Prokaryotic cells are characterized by having no nucleus. I do not have a model of it here. I do not have a model of prokaryotic cell, but they do not have nucleus, right? We agreed on that from beginning of semester. Some of you confused that prokaryotic cells are plant. No, plant cells have nucleus. Plant cells have nucleus. right here. This is a plant cell. This is a plant cell and it has nucleus. It has cell wall. Underneath of cell wall is cell membrane. The yellow is cell membrane, right? The yellow is cell membrane and this one is cell wall. Cell wall, cell membrane. Do you guys see that? Plant cell. Plant cell are rectangular. Generally. Animal cells are what? Generally. They're round. Right? They're round, generally. But there are animals, when you get to zoology first thing you see in zoology, the animal cells are rectangular. But generally, I'm saying generally animal cells, most of the time, 90% of the time, animal cells are round. Okay? So, what they have the prokaryotic cells. That's the mistake I was making my freshman year. I didn't have much biology. I said prokaryote, plants. Wrong. Prokaryotes, it means bacteria, not plant. Plant or eukaryote, they have nucleus. They have nucleus. Plants have nucleus. Kelsey, I'm sorry, you're writing and I'm banging on the table. Sorry about that. Right? Am I making some sense? Okay, DNA in an unbounded region called nucleoid, of course, in prokaryotic cells. The area that DNA is located is called nucleoid. It's not nucleus. You have a quiz on Thursday, on Wednesday. Is that right? Next time we meet. Over what?
Chapter 6.
Wherever I stop. Not entire chapter 6. Wherever we stop on the PowerPoint. Does that make sense? So it would be a nice quiz question. Where are the DNA, the chromosomes of bacteria? In nucleus? No, they do not have nucleus. In nucleoid? Yes, they do have nucleoid. In cell membrane? No, there's no DNA material in cell membrane. Okay? I hope you're going to read your notes. No membrane-bounded organelles. They do not have mitochondria. They have ribosomes, but they do not have mitochondria, or they do not have Golgi apparatus. Eukaryotic cells are more complex because of they have organelles. And I will go over organelles in a minute. Cytoplasm bound by a plasma membrane. Yes, they have cytoplasm. Again, I wish I had a model of a bacteria. I do not. Here is a bacteria. Some bacteria have flagella. Some bacteria have this fimbriae, the hair-looking pilus. I call it pilus. Here is a nice TEM, transmission electron picture of a bacteria. If you're wondering what a transmission electron picture look like this. That's a transmission electron picture. This is a cartoon. I call it a cartoon. Okay, so they do have some, all bacteria have cell wall. The cell wall is different than the cell wall of plants. Structure-wise, a little bit different. Okay, definitely they need ribosome. What is ribosome needed for? Why do they need ribosome?
The structure of proteins.
They make proteins. Ribosomes make proteins. They need that. They need that. Okay, nucleoid, of course, the DNA material, chromosome material is located right here. Plasma membrane, cell wall, capsule. Outside of cell wall, they have a structure called capsule. When you go to, I don't know, you had microbiology or not, gram-negative, gram-positive, capsule, all of that, don't worry about it. And then some of them have flagella. The structure of flagella in bacteria is different. No bacteria have cilia. You never find cilia in any bacteria. But they have flagella. The structure is different than flagella that we know of in eukaryotic cells. And I will talk about flagella in eukaryotic cells, but not in prokaryotic cells. The only animal in human, the only cell that has flagella is sperm. You don't find it anywhere else. You find cilia, but flagella is the only found only in sperm, human sperm. Nucleoid, ribosomes, plasma membrane, capsule, all of those goodies. Eukaryotic cells, on the other hand, eukaryotic cells are characterized by having DNA in a nucleus that is bounded by membranous nuclear envelope. In the models we have, what is the nuclear envelope, or what is the color of nuclear envelope on these? Huh? White. Don't be afraid. If a rank answer is no, I say no. That's not right. So what? If I say no, what will kind of happen? Nothing. So in here, nuclear membrane is white. What color? White. Both on these models. Okay, that's the nuclear membrane, and nuclear membrane is made up of phospholipid. DNA in a nucleus that is bounded by membranous nuclear envelope, membrane-bounded organelles, and then a cytoplasm in the regions between the plasma membrane and nucleus. We already talked about all of those. Eukaryotic cells are generally much larger than prokaryotic cells. Here, let's go over cell membrane, and we'll come back to cell membrane here. We'll come back to cell membrane here in chapter 11, after we are done with chapter 7, I guess, chapter 7 and 11. But keep these in mind that I don't have to repeat these again. The cell membrane is made up of phospholipid, something you already studied, something you already know what phospholipid is. The head of the phospholipid is made up of glycerol and phosphate. Those two. You already know what they are. Glycerol has 3 carbon, and phosphate has a P, and attached to it oxygen. So this is the they call it hydrophilic. What does hydrophilic mean? These are all hydrophilic. What does that mean? They like water. So out here is water, and these heads, which is made up of phosphate and glycerol, they like water. They interact with water. The tail, the 2 fatty acids, either saturated fatty acid or unsaturated fatty acid. You all know the difference between saturated and unsaturated fatty acid. Saturated, unsaturated. They do not like the tail. They call them head and tail. This molecule, just arbitrary, they pick the name head and tail. The tail does not like water. So, when they are arranged, when God or Mother Nature or whatever put them together, they have 2 layers. That's what they call them, phospholipid bilayer. That's what this membrane is called. Phospholipid bi. Bi means what? 2 layer. Hey. Y'all know what I'm talking about. Phospholipid bilayer. So there's 2 layers of it. 1 layer facing outside of the cell. What is outside of the cell most of the time? Oh, anybody? Water. No, water. Water. Outside of the cell is water. What is inside of the cell? Water. Cytoplasm is made up of, it's like a gel, but tell me how much of this is water? You see this gel? How much of it is water? A lot of it. If it was hard, even this one has water, but not as much. It's hard, it's solid. Everything has water. They say black hole doesn't have water. You all know what black hole is?
Yeah.
It sucks everything up.
Most it's water.
Everything has water, but the amount of water in here is not as much as the amount of water in here. Okay? So, inside of the cell you also have water and other goodies.
Yeah.
So, right here the head are facing the water, and the head are facing the water, and the 2 tails the tail of the interior and the tail of the exterior are facing each other. Yes or no? Let's go. Okay, then that's not all of it. You have a membrane, okay? You have a membrane that there are protein molecules, phospholipid bilayer, right? The head, the tail. You all know what I'm talking about. This is the same as this portion. Forget about the other. Just that portion is same as this one, right? Same thing. Outside of the cell, inside of the cell. However, in this diagram, there is one thing missing. What is missing in this diagram? The protein molecules. So cell membrane is phospholipid bilayer. Cell membrane is phospholipid bilayer, and protein molecules are embedded in them. Am I making some sense? A little bit. There are protein molecules embedded in them. The way, the analogy that I make, this is Oreo cookie, right? Look like Oreo cookie. Outside is chocolate, and inside is the creamy part. You guys like Oreo cookie? Not Girl Scout you were eating this morning, and you don't offer any. I mean, do you want some? No, I don't need it, but anyhow, it would be nice to offer. I don't know where it has been.
Oh my God.
But it's a nice gesture. Would you like? It's a respect, I guess. I'm just kidding. Do whatever you want. I'm just kidding. But anyhow, it's like an Oreo cookie. Chocolate, creamy portion, right? Then take this Oreo cookie and put chocolate chip in it. If you patent this, Lauren, if you patent this and you make chocolate chip Oreo cookies, I want part of the wealth. Because I gave you the idea. I gave you, Kelsey, I gave you the idea. It would be nice of you again. It would be mannerism that you knock my door and say, Amir, here's a $2 million from what I patent. Maya, is that right? It would be nice. I made $100 billion. Here is for you, $2 million for you. Say thank you. Oreo cookies with chocolate chips embedded in them. Okay, so you can patent it, make millions of dollars. Here we go. Here is a transmission. Here is a transmission. I've got a picture of it. Okay, 2 layers of phospholipid, and then of course here is the cartoon showing you hydrophilic regions up here, hydrophobic region right here, the yellow, and again hydrophilic region right here, and then right here are protein molecules embedded. Also, continue on, this is not over yet, continue on, attached to these protein molecules, some of them, not all of them, some of them, they have these oligosaccharides. Here is called carbohydrate, but I want you to know it is called oligo. You all know what oligo,
Yeah.
what does that mean?
Small.
Medium, a few. What number did I give you? Right? That's the number I gave you. 3 to 10. 3 to 10 sugar molecules. These are all sugar molecules. Glucose, or other sugar molecules, but mostly are glucose, are attached to the protein molecules. Okay? Lauren, Katrina, there we go. Metabolic requirements set upper limits on the size of the cell membrane. The surface area to volume ratio of a cell is critical. As a cell increases in size, its volume grows proportionally more than its surface area. Here it is comparing the surface area to the volume of these two, if you look at these numbers, you will see at the end of the day that the volume is more on these than these guys. So that's what the surface to volume ratio, is 6. It is 1.2 for that one and it's 6 for that one. You can look at this and enjoy it. We talked about it, not much. I'm not gonna worry about it too much. Here's a cell. Here's a plant cell. It has also cilia or flagella right here, but all of its organelles, which is right here. For your first lab practical exam, you should know the name of these organelles. And you should know for the first exam, next exam, you should know all of these organelles and their functions. Okay? Plant cell, same thing. They have one organelle that animal cells do not have, and that is chloroplast. They both have mitochondria. If you look at it, they both have mitochondria, but animal cells do not have chloroplast. And of course, animal cells do not have cell wall. Animal cells do not have vacuole. A few differences. I hope by end of next week you would know them very well. Okay, I don't know what this is. Let's watch this.
Both animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. When sugars produced by photosynthesis are broken down, their energy is used to make ATP in mitochondria. ATP powers the work of the plant cell. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
Animal cells.
Even if you're just studying, a lot of work is going on in your cells. Let's zoom down and check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, this section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are 2 types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
Did you see that guy? What is the structure of nucleus? Let's go over that. The nucleus contains most of the cell's genes, and I said a few things about it. A gene is a segment of your DNA. Long strand of, she said 2 meters long. I don't know where she's getting that number from, but anyhow, it's a segment of that. Out of that 20 meter, let's say 10 centimeter of it is a gene. That gene is responsible for the color of your hair. So you know what gene is. And it's usually the most conspicuous organelles in the body. So when we look at your slides on the microscopes, most of the cells, when you look at them on the microscope, you see the cell, the nucleus. The other organelles, you will not see it on the microscope. But this one, you do see it. Chloroplast is another one. Another one that you will see on your slides is a chloroplast. So nucleus and chloroplast and vacuole, those are pretty much the most conspicuous one that you can see. The nuclear envelope encloses the nucleus, separating it from cytoplasm. The nuclear membrane is a double membrane. Each membrane consists of a lipid bilayer. Okay, so what am I saying here? That the white structure right here is just like cell membrane. It's 2 layers of phospholipid bilayer instead of 1 layer. Pores, there are pores in here. If you look at it, you see the pores in here, guys. You see the pores in there, the opening. So the nuclear membrane has pores, opening, if you would. The nuclear membrane, the pores regulates the entry and exit of molecules from nucleus. The nuclear size and envelope is lined by nuclear lamina, which is composed of proteins and maintains the shape of the nucleus. And a nuclear lamina is right here, the material inside of the nucleus is the nucleosides, and envelope is lined up by nuclear lamina. That's right here in this area. Here is pretty much the structure of it. I'll let you guys look at it. I'm not going to go over everything in there, but look at it at night. Here are the pores, the protein molecules. These are protein molecules, and the opening is right here, that pore. And then you do have the nuclear pore. This is endoplasmic reticulum. This is the nucleus with the nuclear pore, and inside of it, the chromatin is DNA and protein molecule, right? These are protein molecules, and this is DNA. DNA wraps around the histone, this protein molecule called histone wraps around and goes around another histone, wraps around and goes another histone. Then here is a transmission electron picture of nucleus. So one of the things that I will ask you to know for your lab practical exam is these. I have a notebook here. You're more than welcome to look at it. You are more than welcome to look at these pictures. But you have them. The same thing. Same thing in here. I'll take them out of my binder and put them on the table. What is the name of that structure? What is the name of that organelle? Okay, so that is, make sure you get familiar with these pictures. They're all different. Okay, again, here is the pores. These are transmission electron pictures of the pores, of these pores on the nuclear membrane. And here is a nuclear lamina, which I talked about that inside of the nucleus, that nuclear lamina. Nucleus continues, chromosomes. We human has 46. You don't have to memorize those, but for human, yes, I would expect you to know that. Sheep has 54, cow has 60. I'll look this up online. I used to know them, but I forgot them. But finally, I found the carriage after what, 5-10 years. I put them. Mouse has 40, pigs have 38, horses has 64. So every species is different. The number of the chromosomes from one species to the next is different. But we, all of us in this room, all of us in this room have how many chromosomes? 46. I hope I'm making some sense. So the number of the chromosome in each species is different. The number of chromosomes in fern. Have you guys know what is fern? You know, the plant fern? Fern. Guess how many chromosomes fern has in its nucleus?
100.
300. Very good. You're close. 300. That's a lot. That's a lot. Okay. The DNA and proteins of chromosomes are together called chromatin. Okay. So the DNA and protein molecules, I gave it a name. We called it histone. The nucleolus, she mentioned that when I asked her what is the structure of nucleus, she also mentioned nucleolus. Nucleolus is located within the nucleus and is site for ribosomal RNA synthesis. Ribosomes are the site where ribosomes are being made as nucleolus. What is ribosome? That are made in nucleolus. What is ribosome? Ribosomes are complex made up of ribosomal RNA and protein molecules. You're right. There are two units of protein molecules, smaller unit and large unit. That's what ribosomes are. There are two units of them. Ribosomes carry out protein synthesis. In cytosol-free ribosomes, we have two types of ribosomes. We have two types of ribosomes. One ribosome that is attached to endoplasmic reticulum. You see those white dots? You guys see those white dots? Those white dots are attached to endoplasmic reticulum. And then the other ribosomes, trunk, are free-floating in the cytoplasm. They're free-floating in the cytoplasm. So two types of ribosomes. One that is attached to endoplasmic reticulum, and the other one that is free-floating in the cytoplasm. Okay? Outside of endoplasmic reticulum, a nuclear envelope bounds to ribosomes. Here they are, these little dots. You see those dots? Those are all ribosomes. This is the endoplasmic reticulum. Transmission electron picture again. I'll show you during the exam. You should tell me lab exam, not lecture. Lab exam. You should tell me this is rough endoplasmic reticulum because of these ribosomes. All of these ribosomes on the membrane, this membrane is endoplasmic reticulum, and they're showing you in smaller. And of course, he's showing you the structure of the ribosome is a larger unit protein molecule and small unit protein molecule. Okay. So, and then that's a computer digital picture of it, which I never show you. I never bother you with that one. I never ask you that one. I never show you that picture and say, what is this? If I want to, I'll point a pointer here, a piece of tape. A piece of tape, I will put it on the picture that I showed you in my binder and ask you, what is the name of the structure? That's ribosome. What is the name of the entire page? I will not put any tape anywhere. Just the entire page, that would be endoplasmic reticulum. What is the endomembrane system? Okay, so let's go over that, and then after that, we call off the day. So endomembrane system, and your quiz would be the end of, on Wednesday, all the way to the end of prefix suffixes, microscope questions. What is scanning power? What is low power? What is high power? High power is 40x, is that right? Low power is 10x. Scanning power is 4x. What is total magnification of low power? 100. Total magnification is 100. Your eyepiece is 10. Your objective lens is also 10. 10 times 10 is 100. You're seeing something, you're looking at a cell 100 times bigger. That's what that means. On the scanning power, is total magnification is 40. But what is scanning power is 4x. So study those a little bit. And I've got to give you a handout. I will give you a handout. But anyhow, let's go over endomembrane system. Endomembrane system consists of nuclear envelope. Starts with nuclear envelope. Right here. And then after nuclear envelope is endoplasmic reticulum, right here. You see how they are attached to each other? And nuclear envelope, endoplasmic reticulum. Then Golgi apparatus, right here, these are Golgi apparatus. And then after Golgi apparatus is lysosome. On this model, if I can find a good lysosome, the purplish one, peroxisome is yellow. The purplish one, this lysosome. And then after lysosome, vacuoles. Animal cells do not have vacuoles, but the vacuole would be here. On a plant cell, this big old thing is higher, right here. That's a vacuole. That's huge. And plant cells have it. And of course, at the end is plasma membrane. So it goes from nucleus to endoplasmic reticulum, from rough endoplasmic reticulum to Golgi apparatus. From Golgi apparatus, it goes to
Lysosome.
Lysosome. From lysosome, in this case, animal cell goes to cell membrane. So those are the parts that I was hoping you would know. These components are either continuous or connected together via vesicles. In the plasmic reticulum, biosynthetic factory, in the plasmic reticulum, more than half of the total membrane. In many eukaryotic cells, the ER membrane is continuous with nuclear envelope. There are two distinct regions of ER, smooth ER and rough ER. This is smooth ER. You guys see that? That's endoplasmic reticulum. but it does not have any ribosome. Does not have ribosome. So they call it smooth ER. This is called rough ER because it has ribosome. If it has ribosome, it's called rough ER. It doesn't have, it's called smooth ER. And right here, this is all rough ER. Rough endoplasmic reticulum because of the ribosomes. What are the functions of smooth ER? Quickly, part of, again, you might say, Amir, smooth ER is not part of the endomembrane system. Yes, it is. It is part of the endomembrane system. So smooth ER is part of the endomembrane system as well. Smooth ER, function of it, synthesis of lipids, metabolize carbohydrates, detoxify drugs in liver. If you drank beer yesterday, right now your liver is hard, or alcohol, any kind of alcohol, watching Super Bowl. Right now your liver is hard at work. Working very hard to get rid of all of that toxic material you ate yesterday, or drank yesterday. Okay, and then of course, in muscle cells, it stores calcium. You need, for your muscle contractions, for doing this, you need calcium. And that calcium is stored in your muscle cells. The rough ER, synthesis of glycoproteins and of course protein molecules, distribute, transport, vesicle, secretory proteins surrounded by membrane factory of the cell. These are all of the functions of rough endoplasmic reticulum. Golgi apparatus made up of membranous sac-like cisternae, look like sacs. I'm still talking about the endomembrane system. These are sacs. These are membrane. These have phospholipid, 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 phospholipid. When I say membrane, it means phospholipid. Okay. The Golgi apparatus made up of membrane sacs called cisternae, and then functions of Golgi apparatus, modified products of endoplasmic reticulum, manufactures certain macromolecules, stores and package materials into transport vesicles. So what is the function of Golgi apparatus? Golgi apparatus makes the fold of the protein. You remember I talked about the first, see what this is, the material from first exam will haunt you for next exam. The material for this exam will haunt you, follow you, third exam, all the way to the final. Everything you learn in this class from beginning to the end is connected together. You cannot say, oh, the first material for first exam, I don't have to worry about it anymore. No. You remember what is the name of protein molecule being folded? What was the name of that process called? Renaturation. When you unfold them, it's called denaturation. So all of the protein molecules are being folded right here in Golgi apparatus. Adding carbohydrate to it. Where did you see that today? We talked about it today. Where did you see two protein molecules have carbohydrate? Should I go back on my slides? Where did you see that protein molecules have carbohydrates? Huh? Anyone? Stephen? Do you remember?
Is it the membrane?
Cell membrane. Remember I said Oreo cookies and chocolate chip? In the Oreo cookie you add chocolate chip. Attached to chocolate chip was carbohydrates, right? Oligosaccharides. Where does oligosaccharide attach to protein molecules? Golgi apparatus. Okay, Golgi apparatus. Everything that we say in this class is together, come together. So, stores